Hello, my name's Chris Harris and I'm from Allery Chemistry, and welcome to this video on amino acids, proteins and DNA. Now this video is dedicated to the AQA specification, so if you are studying A-level chemistry and your exam board is AQA, then this is perfect for you. So it's not like maybe that you might have seen other resources, other videos where they may be quite generic. This one is actually dedicated to AQA, so it is designed around the specification. And in fact, there's loads of these types of videos. These are the revision videos. Loads of types of these videos for year 1 and year 2. So it's the full range, all on Allery Chemistry YouTube channel. There's also some whiteboard tutorials for some generic information, some general information on there. And there's also some exam paper walkthrough videos as well, where we go through some variety of different types of exam papers. So they're all on Allery Chemistry. They're all for free. All that I ask is that you hit the subscribe button. That would be fantastic, just to show you support, and also you can you get only any updates of any new videos that go on as well. And as long as people keep watching them, then I will keep watching them, subscribe, and then I'll just keep making them. It's as simple as that, really. This video is actually made from slides that I made for revision purposes, and you can purchase them if you'd like your own copy of them in the link in the description box below. So they're great value for money. You can use them on your smartphone or your tablet, so you can use them for on the move, and scroll through them at your leisure. And you can also, I've known people who print these off and write their notes all over them, to annotate them as part of their revision material. So they're all available there for all the major UK exam boards, not just AQA. There's other ones on there as well. So go and have a look. There's plenty there. Right, so like I say, that this is dedicated to the AQA specification, and so therefore it meets the specification points as it's stated on there. So first of all, we're going to look at amino acids. We're then going to look at proteins. Then we're going to look at enzymes. We're going to look at DNA, and then the last bit is looking at anti-cancer drugs, so things like Cisplatin. So that's the running order for this video. Okay, so let's start with amino acids first. Okay, so amino acids have an amino group, which is NH2, and you might have seen the amines topic already. I've done a video on amines, and the carboxyl group, which is COOH, and I've also done a video on carboxylic acid and derivatives. So if you're not too sure on them, then I recommend you go and have a look at them there. And there isn't a massive amount you need to know regarding the actual the intricacies of amines and carboxylic acids, but you do need to know that one's basic, one's acidic, and you need to know them little bits there. So it might be helpful if you watch them videos first. But this video, like I say, is going to look at amino acids in particular, and it's going to look at these functional groups. So you've got our carboxyl group, and we've got an amino group as well. So an amino acid is basically structured in that way. And we also have an R group as well. Depending on what the amino acid is will depend on what this R group is going to be. But the basic structure is looking a little bit like this. And actually, amino acids are amphoteric. They're a bit strange. They have basic and acidic properties. So it gives us this versatility in terms of the reactions that we can undergo with them. Now, amino acids have an organic side chain, like I say, and it's represented by the R value. That's with the exception of glycine, where R is actually a hydrogen in this case. But any other amino acid apart from glycine, this would be a different group that's attached to it. So it's always this structure. This is called an alpha amino acid because the amine and carboxyl group are actually attached to the same carbon. Don't need to worry too much about that. Okay, so amino acids, except for glycine, they are all chiral. They're all chiral molecules. They have 4 different groups, as you can see, surrounding that central metal ion. So they will actually rotate plane polarized light. And I have done a video on that as well, as you would expect, because the full range is on there, where we look at chirality and we look at optical isomerism and look at how we can detect that. And if you've seen that video, then you'll know that we use plane polarized light to detect chiral molecules. Okay, so amino acids are actually named into in two different ways. They have a common name and they have a systematic name, and this is using the IUPAC rules that you are fully familiar with by naming other molecules. Okay, so you'll see it named too, but what we need to be able to do, the exam board is expecting you to name it using the IUPAC rules as well. So for example, you've seen the word there before, which is glycine. That's the common name, but the systematic name is actually different. So we're going to have a look at an amino acid here now, and we're going to look at how we name it. So the first thing we need to do, this is alanine, that's its common name. So the first thing we need to do is find the longest carbon chain. So in this example, you can see the longest carbon chain is all highlighted in red here. This is 3 carbons, so this is propanoic acid. So remember, if you've seen the carboxylic acid video, carboxylic acid derivatives video, that we always number from carbon 1. So this one here is carbon 1. That's always from the carboxylic acid group. Okay, so that's 1, 2, 3. So there's 3 carbons there. So this is propanoic acid. That's the first one. And then the second step is then we need to number them carbons, like we said before. There you go, 1, 2, 3. So we always number from the carboxyl group. And then what we need to do is then note where the amine group is. This is the NH2 group. So what carbon is that on? Now you can see there that that's on carbon 2. Okay, so we call that amino when we're naming the amino acid. Okay, so we say it's an amino group. And so then we need to name any other groups that may be attached to it as well. So these are anything apart from the amine group that we've obviously taken into account of. So anything such as OH, which would be hydroxy, for an example. So we had an OH group on there. And then finally, we just name it. So the name of this is 2-amino propanoic acid. So propanoic acid, obviously this has priority. We just put propanoic acid, and we just need to know where the other groups are. So 2-amino. So for example, if there's a hydroxy group on there, and that could be, you would put the number hydroxy 2-amino propanoic acid. Okay, so you would always, or the other way around, so you always do it in alphabetical order. So amino is A, so hydroxy would come in between there. So no difference there. You can see the nomenclature of amino acids is not too traumatic. You're just following the standard rules that you'll be familiar with through doing naming molecules all the way through A level. Okay, so amino acids, like we said before early on, that they're actually amphoteric, and so therefore they act as acids and bases. Now, amino acids can also exist as something called Zwitterions, which is somewhere along the line of them being amphoteric. So a Zwitterion is just a molecule with both positive and negative ions. So Zwitterions only exist at the amino acid's isoelectric point. Okay, so Zwitterion, it's actually means double ion, so 2 ions. Okay, so it comes from the German. It comes from Germany. Like a lot of the naming and names you'll see in chemistry, they come from German. So Zwitterion just means 2, so 2 ions. Okay, so if you study German, you'll be familiar with that. So the isoelectric point is the pH at which the average overall charge is 0. Okay, so that's the isoelectric point, and this is dependent on that R group that we've seen before. Okay, so let's have a look at an example of a Zwitterion. So here's your Zwitterion here. So it's likely to be formed at a pH at the isoelectric point. Okay, and both the carboxyl and amino groups are ionized. So you can see here this is a Zwitterion because we've got 2 ions there, a positive ion and we've got a negative ion. So this is likely to be at the isoelectric point, whatever pH that is. Don't assume that this is always at, say, pH 7, because different amino acids have different isoelectric points. Okay, so it really just depends on that. Okay, so let's assume so at low pH, what happens to this ion at low pH? In other words, in acidic solution. So what actually happens is if we put this into acidic solution, we have a high concentration of H+ ions. Now the H+ ions is now going to react with the O- on the carboxyl group there. So it's going to react with that. And so therefore we have OH. Now we have our full carboxylic acid that's put on the air. So this is if the pH is lower than the isoelectric point. This is what we get. So at a higher pH, so in other words, we put it in alkaline solution, then what actually happens is the hydrogen from the NH3 group, you can see here, there it is, okay? So the hydrogen from here drops off, and we form NH2 again, but obviously we still have our negative charge on our carboxyl group, okay? So this is if the pH is higher than the isoelectric point. Okay, so you don't really need to know a lot about that, other than the fact that do exist as Zwitterions. You do need to be aware of, if it's above or below the isoelectric point. What product would you form? So just be prepared to be able to draw the formulas of them. Okay, so that's fine. Okay, so we know a little bit about the properties of amino acids. We know how to name them. We know what they look like. So we know all that part. So now what we've got to be able to do is practically, we've got to be able to maybe identify and separate amino acids, so we know what type of amino acids we have. So one of the ways in which we can do that is using thin layer chromatography, or TLC. So it's a very loving form of chromatography, TLC. So thin layer chromatography allows us to separate, okay, and identify amino acids as they have different solubilities. Okay, so that's quite important because amino acids are actually colorless, and we'll look at how we can detect them later using other methods, but we need to separate them and then identify it. Now this could be useful in terms of identifying amino acids, say proteins in say urine samples for medical reasons, or it might be even if mainly in urine samples, but it could even be in blood samples, etc. It may even be in hair samples, for criminology, etc. So there's a lot of use for this. So this is how, very CSI this. So this is how we're going to identify amino acids using a technique called TLC. So TLC basically uses a stationary phase of silica or alumina, which is this bit here. So this is your plate here. Okay, so this is your stationary phase. So this is not moving. And then what we do, this stationary phase is actually mounted onto a glass or metal plate, which is backed on there to give it some rigidity. And then what we do is you put a pencil line is drawn, as you can see on the plate there. So there's your pencil line, and then we put our drops of our amino acid mixtures on that pencil line there. Okay, so it's very similar to what you've seen a standard chromatography method that you may have done using filter paper. So what we're going to do is place the plate in a solvent. Now the baseline must be above the solvent level. That is absolutely vital. If that solvent level is above the spots that you put on, then they'll just dissolve in the solvent and you won't get any chromatogram. So you must make sure that the solvent level is below that line. Okay. So we're going to leave that, until the solvent has moved up near the top of the plate, and then what we need to do is mark the solvent front after time. So you'll see that what will happen is this this solvent, as you can see here, will then migrate up there, and it will stop somewhere near the top of the line. Well, once it gets to near the top of the line, you whip it out, you whip the chromatography plate out, and then we analyze it. Okay. So it works really by the amino acid spots dissolving in the solvent, okay? But some of them amino acids may not be very soluble and will actually spend a little bit more time or have a stronger adherence to the stationary phase. So what you'll find is you'll get a few dots that's migrating up. So you might get a dot here, and a dot here. So the ones higher up the chromatogram are the ones which are the most soluble in the solvent. The ones which are lower down in the chromatogram are the ones which are the least soluble in the solvent and have actually kind of stuck or spent more time on the actual chromatogram. Okay, so we have our chromatogram at the end, and then what we need to do is basically mark up where these amino acids are, and we use the positions on the chromatogram to help identify what the amino acids are. And actually, we're going to look at the method. Basically, we're going to take this out and look at how we can actually interpret our chromatogram to identify an amino acid. So let's have a look. Okay, so amino acids, like I say, these are colorless compounds. However, they can be seen using iodine or ninhydrin solution or fluorescent dye and UV light. So what you'll see when it actually rises up, you'll not actually see any. It looks as though there's nothing has moved on your chromatogram. So we need to be able to effectively spray a dye on them so we can highlight the spots, and then we can use that to measure the RF value, which we'll look at later in the next slide. And then we use the RF value to work out what the amino acid is, but we'll go through that later. So one way in which we can identify these amino acids is using a fluorescent dye and UV light. So if we add a fluorescent dye to the silica plate, okay, we can actually see this using a UV lamp. Okay, so it's all very high tech. And so the colourless spots on that chromatogram, they will block any glow from the fluorescent dye. Okay, so let's have a look. So it looks a little bit like this. So we spray our chromatogram, and you can see it there, and the whole chromatogram will be the color of the dye. Okay, and then the spots underneath won't actually absorb any of that fluorescent dye. So we shine a UV light on there, and we can see the spots that appear on our chromatogram. So that's one way in which we can see these are our amino acid spots that you can see on the chromatogram there. So that's one way in which we can do it. Another way, which you may have used in school or college, is iodine or a chemical called ninhydrin. So this is a slightly different technique. You've got to be really careful, with ninhydrin in particular. It must be used in a fume cupboard. So if you have used it, you would have used gloves. It's not a very pleasant chemical to handle at all. But what we do is we place that chromatogram in a sealed jar with a few iodine crystals for the iodine reaction. Okay, and basically what happens with the iodine is the iodine vapor starts to stick to the chemicals on the plate, and it's dyeing it purple. Now this can be, as you can see here, let's pull up the picture. So the iodine vapor is known as the locating agent. However, what we can do instead of putting it into a jar, is we can hang this on a bit like a washing line in a fume cupboard, and we can spray ninhydrin solution onto the chromatogram, and we get the same effect. So we don't need to put it into a jar for this. But you can see that instead of doing it this way, where the whole chromatogram is dyed and the amino acid spots are quite colourless, you can see here with this one, it's just highlighting the amino acid spots. And again, we get the same outcome, but there's two different methods of doing it. Okay. Right, so now we've identified where our amino acid spots are and how far they've migrated up that chromatogram. We can now use a little bit of maths to actually work out the amino acids that are present. And we can do that using RF values. So RF values is basically a ratio of how far the solvent has migrated up the chromatogram against how far the spots travelled up. So this is how we can actually identify our amino acids. So you can see the number of spots just before we go into the actual calculation, because this is where we're analysing the chromatogram now. But the number of spots on that plate tells you how many amino acids have made up that mixture. So in other words, if we get on the same, we put one spot on that line, and if that separates into 3 separate spots, then we know that that amino acid mixture is made up of 3 different amino acids. Okay, so the number of spots tells us how many amino acids there are. And the amino acids, like I say, they can be identified by calculating the RF value, and we compare that to a known library of RF values. So here's our chromatogram. We pulled it up here, and we've just color coded it so you can see everything what's going on. So you can see here that we've got our baseline, which is here. There's our baseline, okay, and we've got a spot. We've just simplified this. So you've got a spot here, one spot there, and we've got the solvent frontage there. So that's how far the solvent has actually migrated up that. And you can see we've measured the distance traveled by the spot, and we've measured the distance traveled by the solvent. So the RF value is basically the distance traveled by the spot in red divided by the distance traveled by the solvent in purple. Okay, and if we put them figures in, then we work out our RF value. And what we do with the RF value is we obviously measure that against a known library of sources, and we can do that because it's fixed for each amino acid. So every amino acid has a unique RF value. However, that is only true if we're comparing it against like-for-like sources. So for example, they must have been conducted at the same temperature, the solvent must be the same, and the makeup of that TLC plate must also be the same as well. That also has an impact on the RF value. So you've got to be certain in that data book. You've got to know what conditions did they actually conduct the the chromatogram in, and as long as yours are in the same condition, then you can actually make that comparison and hence use that to work out your amino acid. Okay, so it's all very CSI, like I say. Right, okay, so this is where we're going to get into, obviously there's a bit of biology there, all of this is like the biology topic, like biochemistry, so now we're going to enter into, so if you study biology, you'll probably find this very straightforward, so we're going to enter into a new realm now, so moving from amino acids into something called proteins, okay? Now, if you've seen the polymers topic, you'll know that you need to know about 3 different types of polymer, so, well, 3 different types of condensation polymer, should I say. So the first type was polyamides, second is polyester, and the third one are polypeptides, which are known as proteins. So this topic really goes through this part of the polymers topic. So proteins are polymers, like I say, and they're actually made up of amino acid monomer units. And obviously we've seen some amino acids, so we know what they are. And this is actually, like I say, is a type of condensation polymerization. And so proteins are actually amino acids joined together, and they're joined together with what we call a peptide link. So remember, it's that link when we're joining the 2 monomer units together. Okay, so like I say, if you've seen the other video on polymerization or polymers, then you'll be familiar with this. But like with other condensation polymers, the chain can be broken through hydrolysis, so we can break that down. And remember, that's breaking using water. So hydro meaning water, lysis meaning to break. So when we join 2 amino acids together, we form a dipeptide molecule. Okay, so remember, we had polyesters, polyamides. These are polypeptide molecules. It's basically a protein. So let's have a look. So we've got 2 amino acids here. It doesn't really matter what they are. It's just a generic example. So we've got amino acid 1 and amino acid 2. And so what we're going to do is join these together, and we're going to form our dipeptide here. And we form a peptide link. So a peptide link is your NH, as you can see here, reacting with your carbonyl group. And this is on an amino acid joining together. Okay. Now, because it's a condensation reaction, we actually have water being eliminated. So just like the condensation reactions you may have seen in the previous video on polymers. So let's have a look. So these effectively break away, and it forms a molecule of water, and the bonds that are left behind are then used to then form that peptide link that you can see on there. Now, going forward is a condensation reaction, and going backwards is hydrolysis. So remember, you've seen, you may have seen in the previous video that condensation, we call it condensation because water's been eliminated, but hydrolysis is effectively just the reverse of polymerization. Okay, I strongly urge you to go and see that video if you haven't seen it already, because it'll help with this topic. Okay, so bringing this back again, so the dipeptide has a carboxyl and amine group, you see, at either end for further reactions, and this obviously can then be used to make a polymer chain, exactly the same as any other condensation polymerization reaction. So these groups here can then be broken off, and then you can join more and more links on the side there. So the protein can be broken down into amino acids, so we can go backwards. So this is hydrolysis, remember, but it requires really severe conditions. Okay, so we need 6 mol per dm cubed of hydrochloric acid, which is pretty strong stuff. You want to be really careful with that. 6 molar hydrochloric acid, 110 degrees Celsius, and we need to reflux it for 48 hours. So it takes a lot of severe conditions to break down proteins or protein structures. So they're quite robust polymers. And so to determine the amino acids used to make the protein, we break the bond in the middle of that peptide link. So you can see here, there's our peptide link there, and all we have to do is, if we're working backwards, is we break it there, we cleave it there, and we put a hydrogen on the end here to form NH2 and an OH here to form your your carboxylic acid. Okay, so we break right down the middle of that peptide link. Okay, so, and of course, obviously we add OH and H to each of the amino acid units to reform our polymers, our monomer units that we can see here. So again, this is no different to what you've seen in the other video to do with polymerization, if indeed you have seen it. Okay, so we've looked at the structure of proteins and we know how we make a protein, but proteins are incredibly complex molecules. They don't just exist in one form, okay? They actually exist in multiple forms. So they're actually structured into 4 different levels, and we have primary structures, secondary structures, tertiary structures, and quaternary. Thankfully, you don't need to know all of them. Of course, if you're doing biology, I'm not a biologist, but I assume you probably need to know all of them. But for chemistry, you only need to know the first 3. Okay, so we're going to look at the primary structure of proteins first. Okay, so the primary structure shows the individual sequence of the amino acids that make up that protein. So remember, we said that a protein is a polymer and it's made up of loads of monomer units, which are amino acids. So a primary structure looks a little bit like this. So you can see these blocks represent an amino acid, and they're joined together in a very, very long chain, as you can see there. Okay, so you've got your different groups on the side, and we have our free carboxyl group, as you can see there. So this is then free to join up with another amino acid, and obviously we have a free amine group as well on the end here, and this is free to join up again further. So this is just showing you the primary structure, shows you the individual amino acids that make up that protein molecule. So a protein chain is also known as a polypeptide chain, remember. So each of them black lines on there represent a peptide link. Okay, so they're linking these amino acids together, and that's how we describe a primary structure for proteins. Okay, so secondary, so if you just imagine it, secondary is like we're zooming out a little bit. So we're started at molecular level, okay, looking at an amino acid. We're then zooming out a little bit further, and we can join them amino acids together into a big long chain, and we call that a primary structure. So a secondary structure is zooming out a little bit further, and what happens, a secondary structure is actually the height is looking at the zooming out a little bit further, and we're going to look at the interactions within that. So hydrogen bonds exist between them peptide links in that polymer chain, and this pulls that straight chain that we'd seen there, relatively straight, into a coiled or pleated structure. Okay, so they look a little bit like this. Okay, so them amino acid chains that we've seen there can then be twisted into a nice little pretty shape. So we've got a helix here, as you can see there, or we've got this pleated sheet effect here. So we call this an alpha helix chain and a beta pleated sheet. That's the name that we give to these structures of proteins. Okay, so we're just zooming out a little bit further, and now we can say, right, okay, so we've seen what makes up that protein, and we now can see the shape of these proteins here. Okay, so we're going to zoom out even further, and yet I told you these were complex molecules, aren't they? They're absolutely massive. Right, so now we're going to look at a tertiary structure. And so a tertiary structure is basically where we have them coil chains are then themselves twisted up and form into different shapes. You can see here we've got an example of some of the coils, but the coil itself is then twisted into knots as well. It's a bit like spaghetti in a bowl, to be honest. So you can see here we're just zooming out even further. So it has this unique shape of this protein. This is known as a tertiary protein structure, and the additional bonds hold that long coiled chain together. Okay, so it holds its particular structure. So you can see we've literally started. It's a bit like looking at an individual, then zooming out to look at the town that you live in, then zooming out to look at the country, then zooming out to look at the earth. So it's a little bit like we're going bigger and bigger and bigger. And so this is what proteins look like. Okay, so like I say, you've seen there, proteins have really specific shapes, and they're held together by hydrogen bonds and disulfide bonds. Okay, so this is what proteins are not, although it looks a bit of a mess, proteins do have an ordered structure. Okay, they're shaped in a particular way, and they're held together by these bonds. So we can see here, if we've got our amino acid chain in black, so we've just taken out all the individual amino acids. Okay, so we can see here, this is our amino acid chain. They're all structured together like this. Okay, now what holds this shape together? This is your secondary structure. Okay, your secondary and tertiary for that matter. But this is just showing a secondary structure. Then what happens is we have interactions between some of these molecules that may hang off the amino acids, as you can see there. Okay, and so they have this fixed 3D shape, which is essential for enzymes, which we're going to look at later. But enzymes obviously allow our body to react or undergo chemical reactions with enzymes. Okay, so we wouldn't be alive if we didn't have enzymes, basically. So, for example, breaking down food is a classic example. So there's two types of bonds that hold this protein shape together. So the first one is the purple one there. These are disulfide bonds. And so this is actually created from, so for example, cysteine is an example of an amino acid. It has a thiol group in there, which is SH. So anything with thi is to do with sulfur. I know. So you've got thiol group, which is SH, and they can lose the hydrogen atom, and the sulfur atoms that are remaining can form this disulfide bond, so this SS bond that you can see on there. And the other type of bonding which you'd be more familiar with are hydrogen bonding. So this exists when you have electronegative elements such as oxygen, nitrogen. So you remember that from year 1 chemistry? Yeah. So highly electronegative elements, oxygen, nitrogen, when they bond with hydrogen, that will be on the amino acid group. So you probably have OH groups on your amino acid. And also NH2 groups will work as well because you've got nitrogen, so nitrogen will work there. So you have hydrogen bonding between some of the molecules that hang off there. So this, as you can see, holds this structure into a particular shape. Okay, now if any of these bonds are broken, then the protein structure starts to break down. Okay, so the temperature and pH change the shape of proteins as well, like I say, by affecting the bonding and the formation of disulfide bonds as well. So you might see it. So proteins and structures like this, you'll notice it in things like hair, for example. So if you're straightening hair, hair forms a very specific shape. Hair is protein, forms a specific shape when you put, say, if you put hair straighteners in. So anything like that. I seem to talk a lot about hair to say I don't have much myself, but it does work. So you can see your hair shape, the shape of your hair changes when you apply heat to it. You're effectively altering or realigning that protein shape there. Okay, so there's an example in real life. Okay, so remember we said that these proteins actually have a specific 3D shape, and that's useful in enzymes which help catalyze reactions, okay? So we're going to look at them enzymes in a little bit more detail, okay? So enzymes are proteins and are biological catalysts that speed up a chemical reaction, okay? So they're essential, especially in the body, but they can also be used in chemical processes such as washing powder. So you might have biological washing powder. You can get non-bio and bio washing powder. So your biological washing powder contains enzymes in there. Non-biological or non-bio doesn't contain enzymes. It uses a chemical instead. And some people, biological is generally a little bit cheaper than non-bio, but some people are allergic to the enzymes in biological washing powder and cause irritation. So some people would prefer to use non-bio instead because it doesn't cause that reaction. But so there's the difference. I bet you probably didn't know that. But anyway, so enzymes catalyze metabolic reactions within living organisms, and so they play a vital role in life. So you can see here we've got a picture of an enzyme, okay? An enzyme's got a little bit cut out, and we're going to look at why that is in a minute. So all enzymes are proteins. However, some have non-protein elements as well. Okay, so we've seen what proteins are. So this is looking at an example of how these proteins can form to make something like an enzyme. So substrates, okay, which is this green bit here. So these are molecules that enzymes interact with to help speed up the reaction. So the substrate could be a molecule that you want to try and break down. It could be, for example, it could be glucose molecule from the food that you've eaten, breaking it down into, it'll be a sugar molecule, so a large sugar molecule like a carbohydrate, breaking it down into glucose and into smaller molecules which can then be absorbed into the blood. So that could be an example of a substrate. So enzymes have a 3D active site, which is part of the tertiary protein structure. So remember that big structure there that we'd seen before. And actually this is where the chemical reactions actually occur within the active site of the enzyme. So that's that bit right in the middle there, you can see there. So the substrate would fall into this here, and that allows it to then catalyze the reaction regarding the substrate. So enzymes will only work though with them substrates if they actually fit. Okay, so the active site has a fixed shape, and this is why you can't just have one enzyme, because one enzyme will not do everything. You need a variety of different enzymes within your body, and that enzyme is specialized. It does a particular job. Okay, and so when we say it's specialized, it means it has a very fixed shape. That shape that we'd seen there. So if the substrate doesn't fit in that shape, then it doesn't work as well as it should do. So you can see that enzymes work by receiving the correct shaped substrate into that active site, and they must match for them to be catalyzed, because if they don't, then the reaction won't happen. But the enzymes have chiral centers. So remember what a chiral center is. It's a carbon atom with 4 different groups around it, and you can detect them by shining plain polarized light, and it'll rotate that light. Okay, so this is because they're chiral, because they contain amino acids. And remember, we said that the vast majority of amino acids are chiral. Okay, so you would expect the enzyme to have chiral centers as well. So this means that actually only one enantiomer in the substrate will actually fit that active site. Okay, so they are actually stereospecific. Okay, this is really important, because actually when we're designing drugs, we want drugs to fit particular cells properly. We need a drug, so your enzymes may break that down the drug first, as you may well know, and then the substance, the products of that is then used to then treat whatever it is. Okay, that's effectively what an enzyme does. If you don't have, if for example you've got an enantiomer in there in a drug which doesn't fit that active site, then you're going to be in trouble, because it won't break it down, and it may not be any good in your body. Or it may work the other way, where actually it breaks down that molecule, and it forms a really bad molecule which goes around your body, and that can have a adverse effect as well. So it can work both ways. But let's have a look at the mechanism anyway. We just need to know that it's stereospecific. So you can see we've got our active site, we've got our enzyme, and we've got our substrate. Now the substrate is then going to approach the enzyme, and so the enzyme is then going to lock into the enzyme, and we call this a lock and key model, but I'll come on to that in a minute. So this one fits perfectly, so that's fine. So once it fits in, then reactions can actually occur. So we split that into 2 different products, and we call this a lock and key method, because it fits in a bit like the lock is like the enzyme and the key is like the substrate. Fits in perfectly. It splits into the 2 new products, and that then your body can do whatever it wants with that. Okay, so pretty straightforward. So you shouldn't be too it shouldn't be too tricky. But I think the key thing here is the bit about the enantiomer and being stereospecific. Make sure you're alert to that and you're aware of that. And when they ask a question, just make sure you're aware that not all molecules can fit into the enzyme. It must fit in perfectly, but also they are chiral as well. Okay. Right. So the rate of reaction can be slowed down by using inhibitors, okay, to block the active site from a substrate. So, for example, if we don't want a drug to be effectively released very quickly, we want it to be a slow release one. And what we can do is we can create these inhibitors which block some of the enzymes, and that slows the rate of reaction down. So, an inhibitor is basically a substance that's a similar shape to the active site of the enzyme. But you can see there's our substrate there. So this is in green, and there's our inhibitor here. But you see the inhibitor actually has a slightly different shape, as you can see here. Now what the inhibitor is doing is it's actually blocking the active site here from that substrate to enter, okay? And so the more of these that we've got, the more active sites will be blocked, and therefore the rate of reaction will then decrease. So it really just depends on the amount of these inhibitors that we've got sitting within our active site of our enzyme. So another factor as well to consider is how strongly that actually binds to that active site. So if it binds really poorly, then the rate of reaction won't be reduced as much. So it is about the adhesive power, I suppose, to sit within that active site. That is really very much dependent. If it just falls out, then you've now got an active site that's vacant, and that substrate can then enter at that point. Okay, so you can see that's quite common sense. It's kind of blocked it. It's a little bit like, don't get it confused with this though, a bit like poisoning, so catalytic poisoning. So we're blocking a site from allowing a reaction to occur. So some drugs like antibiotics, these work as inhibitors. Okay, so very, very important this actually, because antibiotics are only be used, and you may know this as well, but antibiotics should only be used if you've got a bacterial infection. So they won't work for viral infections, mainly because of the virus and a bacteria is very, very different. So antibiotics only work if you think you've got a bacterial infection. So some antibiotics work as inhibitors, and what they do is they block the active site of the enzyme that is responsible for making the cell wall of a bacteria cell. Okay, so remember, from a bacteria, it has a cell wall. It has a cell wall. All cells do. They have a cell wall. And if the cell wall can't be made, then the bacteria cell will burst and die. So a lot of these enzymes is basically preventing the production, the tools needed to make the house. It's a bit like, say, if you've got a house and it's going to be built from scratch, then it's a bit like telling the builders that you're not going to get any bricks. They can't build a house, can they? So they can't work if they don't need bricks. If they've got everything else, that's fine, but it's no good if you don't have the outer shell. So as active sites are stereospecific, it can be really difficult to find drugs that fit into that active site. So if the drug's chiral, then only one of the enantiomers will work, and that's what I was talking about before. Sometimes the other enantiomer may actually have a negative effect, on your body. You've got to be careful with that. And so when developing new drugs, it requires a trial and error method, and scientists will try different inhibitors to see which will work and refine the molecule on the ones that don't. Okay, so we're basically trialing it, testing it, trialing it again and testing it. And so you'll know that with any form of drugs must go through a trial phase. Okay, so you manufacture the drug to make sure it actually works, then you trial it on in humans, to make sure to see the impact. Are there any side effects to the drug? Does it actually cure the disease? So there's a long trial process with any development of any drug, and it is fundamental that any drugs that are given to patients are safe to use and actually are effective. They do work, but we need to know how much of this drug do we need. Does it need to be a big tablet or just a tiny one? Does it need to be intravenous, or can it be taken orally, through your gut? And so to speed up the drug development, scientists use computer modeling, to design new drugs to act as inhibitors to fit stereospecific active sites, and the computer models allow us to test how a drug will respond without actually making the drug. And this is brilliant, because scientists, to make a drug, you can make a drug and then trial it, and if it doesn't work, then you think, right, okay, I'll have to make another drug and to make another one. It could take absolutely ages. So using computer modeling, we can design a molecule specifically to fit into that site, then get it out to manufacture, then get it into clinical trials. Okay, so it says it is quite an important process, and you can see there that enzymes play an important role in medicine. Okay, so we're now going to kind of zoom out a little bit further. Okay, so we've looked at enzymes, and we've looked at proteins, and we've looked at how they work. So now we're going to look at something called DNA, which I'm sure you would have heard of. And DNA stands for deoxyribonucleic acid. Okay, so DNA. It's a polymer that's made up of monomers called nucleotides. Okay, so it's another example of a polymer. Okay, so DNA is effectively what makes, it's the instruction manual to make us, to make new cells, to make new new tissues, new people, effectively, So DNA is absolutely vital. Now, a nucleotide is made up of three components. Remember, DNA is made up of a chain of these nucleotides, okay? So we're going to look at the monomer unit, which is the nucleotide, and that itself is made up of a phosphate, a sugar, and a base, okay? So let's look at a phosphate group. So a phosphate looks like that, okay? So it's phosphorus in the middle with two hydroxy groups, as you can see there, two hydroxy groups, and you've got your oxygen groups as well. I've got no minus on there as well. That will come into use later. So a pentose sugar is an example of a sugar. So it looks like that. So that's pentose, or 2-deoxyribose. And then we have bases. Okay, so our bases are adenine, which is A, cytosine, which is C. We have thymine, which is T, and we have guanine, which is G. Okay, and so the nitrogens circled, as you see on there, is where the base bonds with the deoxyribose molecule, okay, which is your pentose sugar. So you can see here, these are the nitrogens that are responsible. So we have 4 different bases. We have one type of sugar, one type of phosphate group, but we have 4 different bases to choose from here, okay? So you don't need to remember the structures of these. You will be given the structures of these molecules here, so don't panic, don't worry too much about this, but you do need to know how they interact within the DNA molecule. Okay, so using them 3 components that we've seen in that previous slide, what we can do is we can come up with 4 nucleotides, okay? So the first one is an adenine nucleotide. So you can see here, just to show you on that molecule, remember we said where the nitrogen, that nitrogen was circled on the previous slide. So that means that this is actually bonding with the pentose sugar, which is here, and then obviously onto the phosphate, which is at the top here. So that there, as you can see, is a nucleotide. Okay, so that's one of the monomers that make up DNA. Okay, but there's some other ones as well, as you've seen. So another one is cytosine nucleotide, and you can see here that the nitrogen here, there it is, that was the one that was circled, that's the one that's bonding to the pentose sugar. And then thymine nucleotide, again, there's the bonding nitrogen. And then guanine nucleotide, there's the bonding nitrogen. So you can see they're all the same. They've all got this pentose sugar and phosphate, and same here. But this one is obviously, it's the base that's changing here. So these are the 4 monomer units, effectively, that make up DNA. And you need to be able to draw the structure of these. But remember, they will give you what they are. They'll show you the formula of them. You just need to be able to put that together. Okay, so these molecules are complicated, and thankfully, because it's chemistry or biology or whatever it is, biochemistry, we can actually simplify this using a diagram. So let's have a look. So we're going to use, it looks like something for first school, doesn't it? So, or primary school. So you've got phosphates, which can be symbolized using a circle. For in this example, it doesn't really matter what shape they are. I've just picked these shapes. So phosphate circle. We've got our sugar, which is a pentose, and our base is given a rectangle. I've just color coded them so we can see the difference. But we can symbolize them as that, so it saves us a lot of time. As you can see, drawing a pentagon is a lot better than drawing a pentose sugar. So actually, what we can then do is we can join them all together. So we can have our adenine, cytosine, thymine, guanine, and that actually builds up. And you can see here, there's our phosphate, which is the there we go, which is the red, and then we've got our pentose sugar, which is here. And you see these join all together, and these form a sugar phosphate backbone with your bases sticking out. You see on there. And so these are obviously, this is our polynucleotide chain. This is what this is. And so the phosphates on one of the nucleotide is covalently bonded with the sugar on another, and this creates this sugar phosphate backbone. So this is fixed. What changes is the bases, or the bases that actually come off the top there. Okay, and so this, you'll now recognize this within a DNA molecule, which we're going to look at here. Okay, so remember that sugar phosphate backbone in DNA is formed via condensation polymerization using nucleotides as the monomer units. Okay, so we're going to use them nucleotides to form this sugar phosphate backbone. So let's have a look. So you can see here we've got our 2 nucleotides here. Okay, so we've got 2 different bases here, and we're going to react them together. So remember, we need to try and create that sugar phosphate backbone that we've seen before. So what happens, you can see here, is that these will join together. These 2 here, these 2 nucleotides will join together to form this, and this is your sugar phosphate backbone, as you've seen before with all the different shapes. And what happens is this is a condensation polymerization. So what happens is the water is eliminated from it, as with any other polymer, and water is produced here, as you can see here. And then as a result, we then form this phosphodiester bond, okay, which is this link here, because water has been eliminated from the reaction. So the OH groups on the phosphate and the sugar, which are these 2, okay, they can react further to extend the polymer chain. So effectively, this OH here can react with another another molecule, and so can this one as well. Okay, so it just joins on. So you can see it's just a type of polymerization. It's a little bit more complicated than the other polymers that you've seen, but DNA is just a polymer, and you just need to be aware that it's a condensation one. So remember, you're removing the water there. So you've just seen it there in that example. That's all you're looking at. Don't worry about the rest of the molecule. Okay. Okay, so DNA, looking at that structure, so we've seen what the sugar phosphate backbone looks like, and we've seen how it's created through condensation polymerization, and we've seen all the bases that are coming off each of them nucleotides. There's a lot of keywords here. So we're going to put all that together to make sense of it all. Okay, so DNA is actually formed from 2 polynucleotide strands. Okay, so remember that. Okay, they're twisted together to form this double helix, and this is your classic shape of your DNA molecule. Now you can see we've got our different just observations on the diagram. You can see that we've got different colors here. So we've got yellow, green, and we've got like a reddish, red, pinky color, and purple. Okay, and you can see these colors represent the bases, and these lines here are your nucleotide strands, your sugar phosphate backbone, and each of them sugar phosphate backbones, you've got bases coming off it. So remember in that previous slide where we showed the diagram and we showed where these bases were coming from, we had our sugar phosphate backbone at the bottom there. Now what we've done is taken a long chain of that and twisted it into this DNA structure that we see here. Okay. So the polynucleotide strands, okay, so these are held together by hydrogen bonds between the bases. So remember them bases sticking out, they actually start to interact with opposing bases on either side, okay. Now, there's a particular order, okay. One base will only interact with another base, so it's effectively best friends, okay. So remember this. So adenine bonds with thymine, which is AT. I remember it as A-team. Okay. So the A-team, if you don't know that, then go and Google it. So I remember it as the A-team. So adenine with thymine is AT, and cytosine bonds with guanine, which is C and G. They're the only 2 which will pair up. C will not bond with T. It doesn't want to be near T. Okay. A and T will only bond together, and C and G will only bond together. So it's they're a bit fussy. They only want to bond in that order. Okay, so both strands in the DNA, as a result, are complementary, which means where there's an adenine on one strand, it will align with thymine, and cytosine will align with guanine. So you can see here that the yellow is matching with the green, so adenine with thymine, so AT, okay, and then C and G, cytosine, guanine. So cytosine and guanine, so they pair up very specifically, and you must remember what pairs with what. Okay, really, really important. So there is a particular order. So it's not just they just bond however they please. That isn't the case. They are a little bit fussy. Okay. So the bases are paired up in specific ways. So we've seen them bases that are formed, as you can see within our DNA, and they're joined together using hydrogen bonding. Okay, so as we've seen before, the A bonds with T and G bonds with C, and the arrangement of atoms, they're arranged in a particular way, and it allows them to actually form a hydrogen bond. So let's have a look. So here's our 2 different bases here. So we're just ignoring the sugar phosphate backbone. We're literally zooming in on them 2 coloured bases that are joining together. Okay, so we've got A and T. So let's have a look at that one first. So you can see, there we are. So the hydrogen bonds form when we've got our delta positive hydrogen, which is here on the amine molecule, which is there. That interacts with an electronegative element, so the hydrogen there interacts with the oxygen on the thymine molecule, which is here, which has got the lone pair, and vice versa. Nitrogen on the adenine molecule has a lone pair of electrons, and that reacts with the hydrogen, you can see, on the NH on the thymine molecule. So you can see here, they align perfectly. It's as though it's been specifically designed. This is sticking out here, and this isn't. So it's perfectly designed, and this means they've got this little interaction here, and that holds them bonds together between the 2 strands in DNA. And so we can also do the same for... here we go. So the A and T form hydrogen bonds, as there's two, like I say, there's two atoms to form hydrogen bonds. And likewise, in G and C, we've got 3 here. So we've got 1 here, 1 here, and 1 here. So remember what criteria we need for hydrogen bonds. So we need nitrogen, oxygen, or fluorine bonding with a hydrogen. So that's from year 1 chemistry. So as long as you're aware of that, then I'm sure you'll be able to find where the hydrogen bonds are in between these two bases. Okay. So no other base pairings can happen, as the partially charged atoms would be too close to each other and they'd repel, or they'd not get close enough to for a hydrogen bond to form. So this is why they don't just pair wherever they want. They are very picky, and so A and T will only bond, and G and C will only bond because they are aligned perfectly to form these interactions. Okay. So DNA is a twisted double helix, and so to allow the bases to align perfectly, to form them bonds. So it's twisted for a reason, because it allows these bases to really kind of align perfectly, and it holds the structure of that DNA double helix that we'd seen before. Okay, so there's a lot of information in there. The key thing is just make sure, okay, run through this video again, go through it and look at them keywords. There's a lot of different keywords, and this is quite tough. Obviously, if you do biology, it might be a little bit easier, but there's a lot of people who don't do biology and study chemistry. So what I'd recommend, particularly for you, for those people, is you just go through this video again, run through it, and just familiarize yourself with all this new terminology. Okay? Right, so let's look at the last, towards the last part of the video, and this is where we're going to look at cisplatin. Okay, so remember we're talking about anti-cancer drugs, and you might have seen this in the transition metals video that I've done as well, where we looked at cisplatin in particular because it's got platinum in there, which is a transition metal. So cisplatin is an anti-cancer drug which has that square planar complex, remember, and it has a platinum metal ion in the middle, 2 ammonia ligands, and 2 chloride ion ligands surrounding it. So if you've seen that video, you'll be familiar with this. And you can see that in cisplatin, cis means the same side, trans is opposite, so it's not transplatin, this is cisplatin. So this means that our chloride ions, as you can see there, are next door to each other, they're adjacent to each other, and our 2 ammonia molecules are also adjacent to each other, so we call this cis. So we have this very specific shape here. Cisplatin is an anti-cancer drug. Transplatin doesn't. Okay, transplatin, if we had their molecules opposite each other, then it wouldn't be an anti-cancer drug. So it's really important it must be structured that way. Okay. So cancer, what is cancer? So cancer is made up of cells that multiply in an uncontrollable fashion. We call that a tumor, and they do this by replicating its DNA. So cancer is basically your own cells. It's your own cells which have copied wrongly. So I imagine it a little bit like, imagine you've got a let's say you've got a textbook, let's say, okay? A textbook has 150 pages, let's say it's got 100 pages in. That textbook is like your DNA. It has all the information that you need to understand whatever that textbook is telling you about, okay? So every day, day to day, your body has to make brand new cells all the time, especially like skin cells, blood cells, loads of different types of cells that you need to replicate. Now, to replicate these cells, we need to make an exact copy of an existing one. So that's a bit like you taking that book and photocopying every page in that book. Now, if you photocopy every single page, you've got an identical copy to your original, and that's fine. But sometimes, because these are happening so often, sometimes there's a mistake. And sometimes you might copy all the pages, but you might forget to copy page 42, in which case there's a bit of information missing. And so you have a cell that looks like a cell, but there's a deformity in that. Now, if that cell, if you then say, right, I've copied that, and then you then take that dodgy copy and copy that one, you're then going to make multiples of these dodgy copies. And so it's a bit like your body does the same thing. It replicates a cell, but this cell's got a slight fault in it. And if that is then copied and copied and copied uncontrollably, so in other words, it's just not stopping, then you form a lump, a growth, and that's a tumor. Now, cancer is dangerous if it spreads. Okay, so if these cells start moving away from the location where it was, then it starts replicating and spreading, then you've got issues, because it effectively needs a blood supply. It's a cell, so it'll take nutrients and the blood supply, and it may push against other organs, which may cause a lot of pain. So it's really, really serious, and it's difficult to treat, because your body, as far as your body is concerned, it's your own cell. So it needs help to destroy, or either mark these cells in some way or destroy them. So cisplatin binds to DNA in these cancer cells, so it seeks out these cancer cells chemically, so it's a chemical drug, and it binds to the DNA in these cells. And so as the complex is attached to the DNA, what it does is it prevents that cell from reproducing through the normal cell division that you would see. So remember, if you do biology, here's the biology bit. So this would be mitosis, okay? So mitosis is where you get the division of cells, and it stops that from happening. The cell dies because it's unable to repair itself, so it blocks the spreading of this cancer. Okay, so it's quite useful, as you can see. So how it works is the chloride ions in the complex are really easy to displace, okay, and they can detach themselves from this complex fairly easily. And so the platinum, so this bit here in the middle, can then bind to the nitrogen atoms and the guanine base within the cancer cell's DNA. Okay, so this can bond quite nicely. These can break off fairly easily. They're quite weak bonds, remember, they're coordinate bonds. So another guanine base can do the same with the second chlorine in the cisplatin as well. Okay, so we've got another base that's in there, so it's a guanine base this time. And so the cisplatin bonds to the DNA. It creates a distortion in that strand, so it creates a bit of a kink in that strand, and it means that it can't unwind itself. So remember, if you do biology, you'll know about RNA, messenger RNA, etc. And the DNA, to replicate itself, it must unzip itself, okay, and it must replicate the bases to form another strand, okay, which is your RNA, and then that is then raveled back up again with another strand to form a new DNA. So if there's a big block, it's a bit like having a book, okay, and somebody has put, you're wanting to copy that book, okay, and somebody's put a hand, say you're turning the pages, you're copying it, somebody's put the hand in the place of one of the pages. It's stopping you from copying all of that. Or let's say they rip out that page. So it's a little bit like this. So cisplatin is acting as that person, putting the hand on that book, stopping you from actually copying it, okay, copying the full amount. So that is effectively what cisplatin does. It really binds on a molecular level to that DNA. The problem is, though, as you may be aware with cancer, is that cisplatin will also prevent healthy cells from reproducing it. It's not as specific as we like it to be, and this is the reason why you still see, even though we can cure the vast majority of cancers, if they're caught early enough, we can. But this is why research still needs to go on, because we develop drugs. So this is chemotherapy, and you see the effects. If anybody's seen somebody going through chemotherapy, you see the effects of, receiving drugs like this. People can lose their hair, for example, because it kills off the hair cells. They can lose a lot of weight. They lose their appetite. They're vomiting. It has there's some nasty side effects with it. But, the advantage, I suppose, is that hopefully it will kill off the cancer and you won't have it anymore. So, that outweighs it's that short-term pain for long-term gain, as they say. So that's the downside. So it affects, like I say, the blood supply. So the immune system is suppressed. So if you're going through chemotherapy, you're probably likely to catch infections where you wouldn't normally carry it because your immune system is down. Obviously, you can risk kidney damage as well, hair cells regeneration. Like I say, you can lose your hair. So, if you're under chemotherapy, you'll be monitored. Your body, you have an MOT regularly to make sure that your body is functioning the right way. Blood tests to make sure that your organs are not shutting down because of this, because of the chemotherapy. So, like I say, the effects or the side effects of cisplatin can be reduced, though, by giving lower doses and using a more targeted delivery of the drug to reduce the attack on healthy cells. Okay, so we use a combination. This is why hospitals use a combination of chemotherapy, which is chemical therapy, and radiotherapy, which is using radiation to shrink the tumour to a much more manageable size, and also you can operate as well. You can physically remove the tumor if it's causing problems. You can physically remove it, but there may still be some cancer cells in there. So it's about trying to blend it with a variety of different techniques to reduce the amount of chemotherapy or the length of time you have to have chemotherapy. Obviously, it depends on how aggressive the cancer is. But like I say, despite the short-term side effects, cisplatin is still used as chemotherapy treatment, and because the long-term benefits obviously outweigh the short-term pain that you may get, because in the hope that it could eliminate the cancer completely. And clearly that's more of a an advantage is despite the side effects, hopefully you'd be cancer-free. Okay, so that's it. So that's the whole video on amino acids, proteins, and DNA. There's a lot in there, especially if you're not au fait with biology and you don't study biology. I recommend you just run through that again and just familiarise yourself with the keywords. There is a full range of AQA videos in year 1 and year 2. There's also whiteboard tutorials and past paper walkthroughs. It's all on Allery Chemistry YouTube channel. It's all for free. All I ask is that you hit the subscribe button. That'll be absolutely fantastic. That'll show you support. And also, you'll get notifications as well. You're keeping up to date with any new videos that I'm putting on. And also, if you want to copy these, click on the description box, the link in the description box, and you'll be able to get a hold of there. They're absolutely great value for money. Right, that's it. Okay, bye-bye.